Hello everyone, my name is Visalhounuo. In this video, I'll be explaining on membrane essence filter and inspissator. Filter works by physically trapping particles larger than the pore size and by retaining somewhat smaller particles via electrostatic attraction of the particles to the filters. Besides porosity, other factors also influence the efficiency of filtration. They are electric charge of the filter or electric charge carried by the organisms or nature of the liquid being filtered. Filtration of liquids is accomplished either by putting the solution through a cellulose acetate or cellulose nitrate membrane with a vacuum, that is, by applying negative pressure in the filter paper, or by forcing the solution through filter paper by imposing positive pressure above the fluid. Filtration of air is accomplished using high-efficiency particulate air filter designed to remove organisms larger than 0.3 micrometer from isolation rooms, operating rooms, or biological safety cabinet. Such filter a filter discs with pores so fine that they will not permit passage of bacteria, as asbestos filter used for removing bacteria. These filters are usually made of asbestos. They are pad-like and thicker than membrane filters. This germ-proof filter is used to replace the Berg-Field biological filter. It has a place in modern pharmacy, and all progressive pharmacists should know what it is and how to use it. The bottom is a circular piece of metal terminating in a metal tube the shape of a chemical funnel and has a removable wire mesh to support the filter disc. The disc is made up of fibrous material packed hard. Next is membrane filter. Membrane filter are thin filters that are made of cellulose. They can be used for sterilization during injection by placing the membrane between the syringe and the needle. Membrane filters have a non-uniform porosity of predetermined size, sufficiently small to trap microorganisms. Using the membrane filter technique, sample is passed through the membrane using a filter funnel and vacuum system. Any organism in the sample are concentrated on the surface of the membrane. Membrane with its trapped bacteria is then placed in a special plate containing a pad saturated with the appropriate medium. The passage of nutrients through the filter during incubation facilitates the growth of organisms in the form of colonies on the upper surface of the membrane. Discrete colonies thus formed can be easily transferred to conformation media. Membrane filter technique is an effective, accepted technique for testing fluid samples from biological contamination. It involves less preparation than many traditional methods and is one of a few methods that will allow the isolation of enumeration of microorganisms. Membrane filters are used excessively in the laboratory and in the industry to sterilize fluid material. Coming to the procedure, collect the sample and make any necessary dilutions. Select the appropriate nutrients or culture medium. Dispense the broth into a sterile petri dish, evenly saturating the absorbent pad. Flame the forceps and remove the membrane from the sterile package. Place the membrane filter into the funnel assembly. Flame the pouring lid of the sample container and pour the sample into the funnel. Turn on the vacuum and allow the sample to draw completely through the filter. Rinse funnel with sterile buffer water. Turn on the vacuum and allow the liquid to draw completely through the filter. Flame the forceps and remove the membrane filter from the funnel. Place the membrane filter into the prepared petri dish. Incubate at the proper temperature and for the appropriate time period. Count and confirm the colonies and report the result. Advantage of this membrane filter technique is it permits testing of large sample volumes. Theoretically, almost any volume of non-turbid water could be fed through the disc. The organism from any different any given volume being deposited in the disc. The membrane can be transferred from one medium to another to purpose of selection or differentiation of organisms, thus allowing isolation and enumeration of discrete colonies by bacteria. Results can be obtained more rapidly than the conventional standard method. It provides presence or absence information within 24 hours. Uses of membrane filter. Membrane filters are used extensively in the laboratory and in the industry to sterilize heat-labile fluid materials. Effective and acceptable technique to monitor drinking water. And then it is useful for bacterial monitoring in the pharmaceutical, cosmetic, electronics, and food and beverage industries. Allows for removal of bacteriostatic or bactericidal agent that would not be removed in pour plate, spread plate, or MPN technique. Advantage of filtration sterilization is less capital intensive, suitable for heat sensitive liquids, and large volume of liquid can be filtered reasonably fast. Limitation of filtration sterilization are only liquids and gases can be sterilized by this process. Filters are exposed to replace especially non-filters. Inherent limitations of material used in filter affect the efficacy of this process. That is, breakage of glass filter, rupture of the membrane, and absorption of the filter by Seitz filter. And then next is clogging may occur. Next is Inspissator. Inspissator is a convenient and effective system designed to produce large batches of uniform culture medium 4 to 6 times per day. Vessel containing culture medium and the incubated on a show tray, which is connected with water held at a constant temperature 50 degrees Celsius or 85 degrees Celsius with a tank, so ensuring that the temperature of the vessel is constant. Inspissator takes 50 minutes at 85 degrees Celsius, standard temperature 85 degrees Celsius, operating temperature range ambient plus 55 to 90 degrees Celsius. Capacity for up to 156 test tube or 162 universal container. Sterilization. Process of heating an article on 3 consecutive days at 80 to 85 degrees Celsius for 3 months. The method kills all forms of vegetative forms of spores. Coming to the uses, sterilization of media, example Lowenson Jensen and Loeffler render sterile at 80 to 85 degrees Celsius for half hour on 3 consecutive days inspector. And then next is tuberculum bacteria culture. Coming to the component and operation, Inspissator is built of rust-proof stainless steel sheet. Inside chamber, body and water bath made of stainless steel. Inside chamber 25 degrees Celsius into 21 inch. Outside body 29 inch, 25 inch and 52 inch with heavy duty wheels. Inside the Inspissator, 3 stainless steel ware trays are provided to hold media bottle. These trays are removed from main India for easy lodging of media bottles. Trays are positioned at an angle of 20 degree approximately to provide slope to media. Inspissating capacity of the Inspissator or of McCartney media bottle to about 450 bottles in 3 trays with single layer of bottles in early trays. Capacity can be increased 600 by losing more bottles in each tray in the 2 layer or in the 3 layers. Bottles are at angle of about 20 degrees Celsius. Each Inspissator has 2 water heater, 1.5 kilowatt each, running at 220 voltage. Two spare heaters are additionally provided for emergency use in case of failure of one of the working heater during the working of the inspissator. Temperature range of the serum inspissator is from 5C above ambient temperature to 95 degrees Celsius. Temperature is settled by the user. Accuracy of inner temperature if is better than 2 degrees Celsius. Least count of controller is 1C. Sensor used in patient treated.